Welcome to Health Matters. I'm Euan Ashley, a professor of cardiology and genetics at Stanford. And I'd like to tell you over the next few minutes some stories from the work that we've done here at Stanford over the last 10 years relating to the human genome. These stories are to be found in my recent book, The Genome Odyssey, Medical Mysteries and the Incredible Quest to Solve Them. The first question, though, is who is this guy and where did he come from? And much more importantly, what is he wearing in this photo? This is the Scottish national dress that you might recognize. This is the Fraser tartan, and this is my kilt and sporran. I don't wear this very much in California. Back in Scotland, however, we wear it quite a lot. This is a picture of my homeland. I didn't actually grow up in the Lafroig distillery on the west coast of Scotland, on the island of Islay. I grew up in Glasgow, a major city in the mainland of Scotland. But this is a place we visited and a place that makes very nice single malt scotch that you might like to try if you haven't. This is what counts for a swimming pool that we would jump in the summer in Scotland. This is me and my dad from many years ago. This is me shivering, doing some fishing back in Scotland around 7 years old. And I realized from a pretty early age that I was a bit of a nerd. I loved the Rubik's Cube. I loved to program computers. I wrote a tax program for my dad's medical practice when I was 12 years old and realized that the future was in data. I always wanted to be a doctor, though. This is a picture of my father, who was a local GP, and my mother, who was a midwife. And they used to take me out on their house calls. And so I learned from a very, very early age that medicine was about much more than a job, more than even really a profession. It was really a way of life. I trained in medicine at Glasgow in Scotland and moved to Oxford to do a PhD in molecular genetics, and then took a giant leap across the ocean from the UK to Stanford, California in 2002, where I came for, as I thought at the time, 2 years. 20 years later, I'm still here, and I'm incredibly grateful to Stanford campus and to Silicon Valley in general for providing me incredible opportunities. In particular, I've had the opportunity to spend time with leaders of the governments of the United States and the UK and many other places around the world, some of the leaders of Silicon Valley and other technology companies. And I've started a family here, which I'm very happy to say is there are 3 young kids who are now healthy and living their own lives here in California. This is me in my work clothes. I'm an attending cardiologist at Stanford Hospital where I see patients with heart attacks and heart failure and heart transplants. But in my lab, that's where we've spent time thinking really hard about the human genome. The human genome is a secret code that is inside almost every single cell in your body. It's a code that's unique to you. It connects you to every other living organism on Earth. It connects you to the entire history of the human race. It's 6 billion base pairs, 6 billion data points. Each one of them is an A, a T, a G, or a C. And if I was to take out the genome from every single cell in your body and stretch it end to end, it would reach to the moon and back thousands of times. In fact, if you were to stare at this screen continuously for 24 hours a day, 7 days a week, it would take you 3 months before you saw every single base pair in your human genome. So not surprisingly, to sequence the genome of just one person, the Human Genome Project, took 10 years, 10 countries, and multiple billions of dollars. Between 1990 and 2000, when this New York Times article declared that the genetic code of human life was cracked by scientists, the draft sequence of the human genome was released to the world, a huge amount of blood, sweat, and tears was expended. The next genome was that of the person you also saw on that page, Craig Venter, next to Francis Collins. And his genome cost $100 million. And over the course of the next 10 years, the cost of genome sequencing dropped at such a fast rate, it was compared to Moore's Law. Gordon Moore was the co-founder of Intel, the semiconductor company, and whose products really pushed the technology of Silicon Valley forward. In fact, the technology of the transistors on an integrated circuit were moving forward so fast that it doubled every 18 months. And this became emblematic of the idea of technology advancement. But as you can see from the graph, in 2008, the cost of genome sequencing left Moore's Law in the dirt. You can now sequence a human genome for $500. And that's just an unprecedented change, an unprecedented move forward of technology over the course of just 10 years. I often show this graph, but I find that people remember much more clearly when I put it in Ferrari terms. Now, your average Stanford faculty member like me isn't able to afford a Ferrari like this. I used to drive past this one on the way home from Stanford from my lab. It comes in at around $400,000. But if this Ferrari had dropped in cost as much as human genome sequencing had dropped in cost, that Ferrari could be yours for 1 cent. 1 cent. 10 years later, and this technology is available for everyone. And that story of how we got from one genome to millions of genomes is one I'd like to tell you just a little bit about over the course of the next few minutes. The first genome I looked at was that of a friend and colleague, Steve Quake, shown here on the screen. He's the chair of bioengineering at Stanford, and I wandered into his office in late 2009, and I was there to talk to him about a genetics seminar. But we never really quite got to that, because when I found his office in the curiously labeled buildings of the Clark Building here on Stanford campus, I found him huddled at his computer pecking at the keys, and on screen were a whole series of A's, T's, C's, G's. Basically, his human genome was on the screen. And I realized in front of me was one of the only people in the world at the time. In fact, one of only 5 people in the world to ever have his genome sequenced. He had made headlines around the world that summer when, with 2 people in his lab and over the course of 1 week, for a cost of $50,000, he had sequenced his entire genome. Now, I've told you a genome costs $500. You think $50,000 is expensive, but the one just before Steve's cost $200,000. So this was a major advance. But it wasn't so much the ability to sequence the genome as what was in it that really counted. And as he started to show me variants he had in genes that I knew well because of my work as a cardiologist at Stanford Hospital, I recognized that he had variants in genes that could potentially cause heart disease. And I started asking him about his family history of heart disease. It's shown here. Anywhere you see orange on this family pedigree is someone in Steve's family who has suffered a form of heart disease. In particular, he showed me variants in genes that can be associated with diseases that can cause sudden death. And I asked him if anyone in his family had ever died suddenly, and his cousin's son, shown on the bottom left of the screen here, died aged 19, having never had a sick day in his life. And at that moment, it became clear that Steve wasn't just about to be a colleague that I had at Stanford, but he was about to be my patient. And as I invited him to clinic that day, I realized he was about to be the first patient anywhere in the world to walk into a clinic with his genome on a hard drive. And much more scarily, that clinic was mine. We were in a situation in medicine where we were used to looking at studies where there were 5 numbers, a cholesterol panel. Suddenly, we had a genetic test, a molecular test, a medical test that had 6 billion numbers in it. How were we going to make sense of that, especially in a 12-minute doctor's appointment? So we set out over the course of the next year to build tools that would allow us to understand the genome in the context of medicine for the first time. We built tools like the one you see on the left, a scorecard for a number of different diseases, where our tool can read across your whole genome and push the needle in one direction or the other, increasing or reducing your probability of suffering these diseases. We integrated your genes with your environment. It's very important to be able to say what risks do you carry in your genome that when exposed to the right environment would actually manifest as disease. And we published our paper at the time, and we're very happy with the work we did. But I don't think we were really ready quite for the reaction it received. And over the course of several weeks around the time when the paper came out, we were deluged with interest. Something about the human genome, something about staring deeply into somebody's human genetic makeup struck a chord with people. And the idea that people were really ready for an individualized or personalized form of medicine. And we spent time talking to reporters in many different countries in many different times. Over the course of the next few years, we were lucky enough to spend time working with the Obama administration and others, helping advise on genomic medicine, or as we more commonly refer to it now as precision medicine. Well, shortly after that, I received a phone call from a father of 2, shown here actually, John West. And he started asking me about a pulmonary embolism that he'd suffered. This is a clot that usually starts in the legs, that travels up to the heart and then into the lungs, and it embeds, causing pain and shortness of breath. And as he started to describe this to me, I thought it sounded very much like he had a clotting problem. And so maybe he should talk to a hematologist. But before I got to that, he mentioned that he had sequenced the genome of his entire family. And he'd seen the work we did with Steve Quake, and he was wondering if we could help him work out what was happening in his family's genomes, and in particular in his genome, to predispose him to a pulmonary embolism. Well, that took us to an unusual place. And I don't know if any of you in the audience are from Buffalo in New York or are listening to this talk from Buffalo in New York, but the only thing I knew about Buffalo in New York before this study was that's where chicken wings came from. And actually, the story of where chicken wings came from is pretty interesting. And if you're interested, you will find it in the book, The Genome Odyssey. I won't tell you it today. But another thing that happened, and another thing that came from Buffalo in New York, was the human genome reference sequence. That first genome that I mentioned at $3 billion came actually from 10 people. who had their blood drawn in Buffalo in New York, and that blood was then made into DNA and essentially put in a pool and sequenced as part of the Human Genome Project. Because they were real people, though, it turned out one of them actually had a genetic variant that predisposed to clotting, which we found when we went to look in John West's genome for the cause of the clotting that caused his pulmonary embolism. And it led us to build a whole new series of tools, just like this one shown here, that help us understand the genome in the context of our family. We share so many things with our family members, the environment we grow up in, towels, toothbrushes. We also share DNA. And this tool allowed us for the first time to manage to map pieces of DNA from each individual within this nuclear family, 2 parents and 2 kids, so that we could understand which genetic variants came from mom, came from dad, or were brand new in the individual kids, to help explain their risk for numbers of different diseases. So we started moving genetics into a world where it was the family that mattered. We also, this being Stanford, started a company to help us deal with all the incoming requests we had now for examining the genomes of individuals to understand their medical risks. And so I'd like to tell you over the next few minutes a couple of stories from 2 patients that are very dear to our hearts, where we used the human genome to illuminate their medical future. The first one was a little baby who first came to our attention when she was 36 weeks. That's 36 weeks of gestation. She was still swimming around inside her mom. And she was found to have a heart rate of 70 beats per minute, which is okay if you're an adult sitting in a chair. But if you're a little girl swimming around in your mother's womb, your heart rate should be more like 140 beats per minute. So we brought mom and baby to Stanford, where the baby was delivered by cesarean section. And we found the reason for the low heart rate was actually a genetic condition called long QT syndrome. QT is the region of the heartbeat where the heart resets ready for the next one. And this explained why her heart rate was so low. But the low heart rate wasn't so much the problem as the fact that she was predisposed to potentially fatal cardiac rhythms. And on the first day of her little life, she suffered 5 cardiac arrests. And 5 times, the team at the bedside performed CPR to bring her back to life. We knew early on that this was a very severe presentation and that we had to do everything we could to save her life. She became the youngest baby in history to have a defibrillator placed in her abdomen. This is a device that can deliver a shock to bring the heart back to normal rhythm. But we also knew with this genetic condition, if we could understand the underlying molecular biology, the actual gene involved, we could give her a therapy that was tailored to that exact underlying condition and give her the best chance of survival. At the time, genome sequencing was just present, but it often would take months and months in large teams. If we even did clinical sequencing through the regular lab, it would take 3 months. It would sequence 5 genes, and it would cost $5,000. This little one didn't have that kind of time. So we pulled every thread we could. We spoke to every collaborator we could, and over the course of a weekend, working with Illumina, the sequencing company in San Diego, we managed to sequence her genome over the course of 2 days. While those 2 days were going past, a postdoctoral scholar in my lab, James Priest, worked on our computer methods to speed them up so that we could get an answer even quicker. And in the course of just another day, we managed to work out that this was a particular form of long QT syndrome caused by a potassium channel. This allowed us to let the bedside team know immediately the right treatment to give at the right time and the right treatments that they could pull back on for her ultimate survival. We were really happy to see her 6 months later at a little park just outside her home, and I recently had the chance to see a picture of her from her 5th birthday party. She's doing incredibly well, really without any idea, for the most part, of the technology that was applied to the first few hours and days of her life. Well, in her case, we understood what condition she had. We did an electrocardiogram, and we realized what condition she was suffering from. Many patients go from doctor to doctor to doctor, accumulating mountains of paperwork, mountains of both emotional and financial burden, without ever having a diagnosis. This is often called a medical Odyssey, after the classic Greek poem from Homer called the Odyssey. You'll notice it says on the front cover this was voted one of the best books of 1995. Including, actually, if you look on Amazon, you can see you can even follow the author, Homer, who probably isn't doing any more writing. These medical odysseys, though, are what we attack in a particular consortium of people called the Undiagnosed Diseases Network. And this is a group of doctors across the country and even with roots internationally, where we take on the hardest cases in medicine. And the armamentarium that we have Well, first of all, we use a Sherlock Holmes type approach to try to bring the smartest people into the room, to try to touch everyone we know to try to find an answer. But when that fails, and it's only successful about 10% of the time, we have to bring in the big guns. And in this case, the big guns are the genome. And that's what we did for these two boys. This is Carson and Chase Miller. Carson was the older of the two brothers, and he was born and hit all his milestones around the time of the first year. But as he just became 1 year old, he started to step back a little bit. And whereas he'd previously been cruising, almost walking and feeding himself, he was no longer able to do that. When his brother Chase came along and also didn't manage to hit the same motor milestones, we realized this, his dad, Danny, realized this must be a genetic condition. And he, with his mom, Nikki, started taking the boys to doctor after doctor after doctor. They went from specialist to subspecialist to super specialist to multiple neurologists, and nobody had an answer. They even had a particularly fancy genetic sequencing where you sequence all the genes in the genome, called an exome, and that still didn't find the answer. At that point, they were referred to the Undiagnosed Diseases Network, this group of doctors I mentioned. And we saw them at Stanford. And the first thing we did was sequence the genomes of all 4 individuals, mom, dad, and the 2 brothers. And the power of having the family's genomes together, like I just mentioned for the West family a few minutes ago, we were able to bring that to bear to make a diagnosis and to be able to work out that Carson and Chase were the 8th and 9th boys in the world ever to be diagnosed with a syndrome called MEPAN syndrome. Now, I won't be asking difficult questions about this biochemical pathway at the end of the talk. The details don't matter so much. But the MECR gene that you can see at the bottom right was disrupted in both boys, and it explained their inability to meet their motor milestones while remaining cognitively intact. This was a characteristic seen across all the boys who presented with MEPAN syndrome. Now, if you imagine this biochemical pathway is like a little factory inside each of the cells, then the particular item is taken by one factory worker, they apply their particular, if it's, let's say it's a factory of dolls, they'll apply perhaps the top or the bottoms, and they'll pass it on to the next one in line. But if you take one factory worker out of the line, then of course there's a buildup of those dolls. There's nothing that happens downstream. What you really need is a genetic therapy to replace that factory worker, a way of just putting someone who knows exactly what to do and when right in at the right spot at the right time. That holy grail of genetic therapy is not yet with us, but it is the future. But right now, if you were able to replace some of the elements downstream, let's say fully clothed dolls further down the factory floor, and in this case, that would be something called lipoic acid, then you might be able to help significantly. And right now, if you're thinking lipoic acid might be some incredibly expensive therapy that would bankrupt our healthcare system, you'd be wrong. Because just a few clicks away, when Danny was spending hours into the middle of the night trying to work out what was up with his 2 boys, he was able, after we had the diagnosis, to find lipoic acid on the internet from Amazon for $16 a bottle. Literally the answer, part of the answer to his boys' significant, devastating neurological disease was to be found just a few clicks away. That's the power that a diagnosis can bring to these families, the power of understanding the starting point and on the route towards a cure. And here's a picture of Danny with his 2 boys. They were kind enough to appear on CBS this morning when we released the first results from our study from the Undiagnosed Diseases Network. We released data on the first 1,200 cases. We solved 132, about 35%, and defined 31 new syndromes, all through the power of the genome. In a year when there was a lot of other bad news, we were delighted to be featured as This Week in Good News in the New York Times. Well, rare disease is where the genome has been. Where is it going to go going forward? What about the diseases that many of us who are a little older than those kids that I told you about there are suffering? What about heart attacks and strokes and cancer? Well, the genome can be very effective there as well. In fact, we're moving in our use of the genome from a reactive approach where we try to diagnose after the fact to one where we move forward to be proactive in preventing disease. And nowhere is that more important than preventing heart attack. If you go to your general practitioner and talk about your risk for heart attack, he or she will ask you about smoking or diabetes or cholesterol, and he or she will then put those answers into a calculator to tell you what your risk will be and help you decide whether to take aspirin or a statin medication or do other preventive health measures like diet and exercise, which everyone should do, by the way. But if you were to add the nature component to the nurture that you were just asked about, you would have significantly improved ability to predict that risk. As shown here, the predictive power of a genetic risk score exceeds the predictive power of all those questions you were just asked. But of course, the answer is to put the two together. And I'm very happy to say that at Stanford this year, we're launching a pilot of preventive genomics, where we use sequencing of the genome to be able to provide both the nature and the nurture component to predict risk of heart attack and cancer, so that the power of the genome can come to everyone. Well, in the last few minutes, I want to tell you about another genome. And here it is on the screen. In fact, this is the entire genome. It doesn't take you 3 months of watching a screen to see it all. This is in fact the protein sequence for the particular genome in question that is 30,000 letters, much smaller than the 6 billion in your human genome. But don't let its size fool you, because this genome brought the world to its knees. This is the genome of the SARS-CoV-2, the novel coronavirus responsible for the pandemic. And when it was sequenced and the Chinese Health Authority released that sequence on January the 10th in 2020, within hours we were able to build tests across the world for infection with this disease. The power of the genome was such that we had those molecular tests ready almost immediately. We were able to do that at an individual level, and many of you may have had a rather uncomfortable nasal swab or a saliva-based test for SARS-CoV-2. Also, what we're able to do is have an early warning system. Here, we can pick up the SARS-CoV-2 genome from the wastewater in San Jose, predicting 7 days in advance if there was going to be a spike, and allowing us to prepare the appropriate ICU beds and healthcare resources. If we'd moved a bit faster, perhaps we could have moved technologies like this that we're now supporting at Stanford forward quicker. We will be ready for the next pandemic with tests that are $1, 30-minute saliva tests that you can do at home. If this technology invented by Manu Prakash from Stanford was available in the early days of the pandemic, we would not have needed lockdowns. We have the technology, and it's genome-based technology. The genome tells us other things. It tells us about the variants in the virus so that we can track the virus in real time through the world and make sure our treatments are focused on the right variants. It also reveals health disparities. And in particular, in this work that we published here from Stanford patients, where we sequenced both the virus and the human who was infected, we realized that those underrepresented in society were those who were overrepresented in those coming forward infected with SARS-CoV-2. The genome can shine a light on those disparities and help us make healthcare more even, make sure we're looking after everyone in society. But the biggest effect of understanding the genome of the novel coronavirus was the ability to develop in days, albeit building on decades of scientific work, but to build in days genomic vaccines. Each of the major vaccines that is now in use around the world and that is showing us the exit door to the pandemic was developed as a result of that sequencing of SARS-CoV-2, in many cases through academic and industry collaboration. And this was genomic vaccine development at earth-shattering speed. This used to take 5 years. This was done in 9 or 10 months to take us from a point where we had no exit to the point where we can clearly see where we need to go. To prevent the next big one, we need a global biosurveillance force. We need to get ahead. We can guess what the next cause of a likely pandemic would be. We can get ahead of it, sequence it, even build vaccines for it in advance so that we're ready if one hits. Genomics has brought us to the point where we can apply to infectious disease the sorts of revelations I've been able to share with you that we've applied in rare disease, and the preventive genomics we're looking forward to do over the next few years for common diseases like heart attack. It's been my pleasure spending a few moments with you. If you've enjoyed hearing these stories, I encourage you to get a hold of the book, The Genome Odyssey, and learn about more medical mysteries and the incredible quest to solve them. Thanks so much for joining me today.